Hello students, welcome back to a new video of Sunanda's tutorial. Today we will discuss about the Telome theory. So according to a general belief that the earliest and primitive sporophyte of land plants was axial in nature, that means it consists of a single axis. The leaves and branches were evolved later by modification of central stem. The primitive land plant were supposed to have rootless, leafless and dichotomously branched green stem. Dichotomously branched means like this. The vascular system was simple protostele. Protostele is the simplest type of stele found in plants. Some of the ultimate branches terminated in sporangia. These assumptions are supported by discovery of some simple plants in Upper Silurian and Devonian period of Earth's history like Rhynia, Horneophyton, Xylophyton, Asteroxylon and others. Discovery of such plants led the botanists to propose theories explaining appearance of leaves, branches, roots and sporangia. These theories tend to explain the process of elaboration of primitive sporophyte into modern sporophyte possessing roots, leaf and variously disposed sporangia. That means the botanist thought that the earliest plant was a simple axis or a dichotomy with a dichotomy, but they lack leaf, root and stem. Later on, the modern sporophyte have roots, leaves and stem. So there are many theories who are developed. There are many theories developed by many scientists to explain this modification or this elaboration or this evolution. Many theories are proposed. Among these theories, the Telome theory proposed by Zimmermann in 1930 has attracted great attention and has a large number of supporters. Telome theory was first proposed by Zimmermann in 1930 and elaborated by him in 1938. He concluded that the vascular plants evolved from a simple, leafless, dichotomously branched ancestral type like Rhynia. Such plant is made up of sterile and fertile branches. Some of the branches are sterile but some are fertile, and the fertile branches ends with the sporangia. The simple and terminal part of dichotomously branched stem were designated as telomes. So these are telomes. These are telomes. The terminated part are called telomes. The telomes meet each other. The telomes meet each other at the point of forking. At the point of forking, portion of the stem below the forking were called mesome. So these are mesomes. The telomes were classified as sterile telome or vegetative telome called as phylloid. The telomes which lack sporangia are called phylloids and which bear sporangia are called fertile telomes. The fertile telomes consist of a terminal sporangium usually traversed by a single vascular strand up to the base. That means sporangia have vascular supply. As evolution proceeded, there was a change from dichotomous to sympodial branching as a result of fusion of two or more telome called telome truss. These telomes fuse with each other to form syntelome. The fertile telome trusses are called sporangial trusses and sterile telome trusses are called phylloid trusses. Both the types of syntelome were occurred together to form mixed telome trusses or mixed syntelome. So these are fertile telomes with sporangia and these are sterile telomes without sporangia. Then the process of telome theory. According to the Zimmermann, this telome process of primitive Rhynia type of vascular plants have been subjected to certain evolutionary processes in varying degrees among the various taxonomic group. So they followed some steps in the evolutionary process to become the modern sporophyte from primitive type. So they followed these steps like overtopping, second planation, then syngenesis or webbing, then reduction, then recurving. At first, the overtopping, the equally dichotomizing axis developed unequal dichotomy. This results in the formation of long and short branches. Some branches are short like this and some are long. The short branches appear as lateral shoot. This led to development of a sympodial axis, which ultimately changed to monopodial axis with lateral branches. These lateral branches later on metamorphosed into leaves. Then planation. The equal dichotomies were originally in more than one plane. They were arranged in planes successively at right angles. In the step of planation, the unequal dichotomies that are placed in more than one plane were placed in a single plane in the process of planation. During the process of planation, the dichotomies become arranged in a single plane. It is an important process that led to the evolution of leaf. Then the third step, which is syngenesis or webbing. As the name suggests, the adjacent telomes or mesomes were connected with each other by development of parenchymatous tissues between them. This is also called as parenchymatous webbing. During this process, stelae of the concerned telomes were fused, and syngenesis considered to be two types by Zimmermann. One is foliar syngenesis, one is axial syngenesis. Foliar syngenesis, during this process, there is coalescence or fusion of apical meristem of telomes, where this meristem fuse to form a marginal meristem. A lamina with veins develops. The activity of this meristem led to the formation of parenchymatous webs like this between the telomes. Such developments resulted in the formation of lateral foliar appendages. Then axial syngenesis. Zimmermann visualized that during this process there was absorption of a number of branches into single stout axis. Many branches into single stout axis. The fusing branches had protostele, and their fusion led to appearance of a complex organization. Previously, these branches have protostele, that is simplest type of stele. Stele means they have xylem surrounded by phloem. So this is the simplest type of stellar organization, which is known as protostele. But after syngenesis, a complex stellar organization was observed. In the case of shoot united by formation of parenchymatous web, the resulting stellar organization is a polystele condition. That means they bear many stele or vascular bundles. In case during axial syngenesis, the steles anastomosed in different manner. The resultant stellar organization was of diverse type. For example, siphonostele, eustele, solenostele. I will explain this type of stele in my next videos. Then reduction. It was supposed to have brought about by evolution of simple unbranched microphylous leaves of lycopod. It was brought about by reduction of syntelome to a single needle-like lateral appendages. Simple needle-like lateral appendages. in case of lycopods. Then recurving. In this process, the fertile telomes were supposed to become reflexed. As a result, sporangium assumes an inverted position. They assumes in an inverted position. Zimmermann called this process as incurvation. This process is called as incurvation. Wilson recognized two processes. One is recurvation. During this process, sporangia bent downward as in Sphenophyta, and incurvation, this led to shifting of sporangia to ventral surface of the foliar appendages, thus bringing about the condition found in ferns. So, into ventral surface, foliar appendages on the ventral surface of foliar appendages, which is known as incurvation. And in recurvation, they bent downward like this. So it is recurvation. So it is these are the steps of your Telome theory, and it is all about Telome theory. In my next class, I will explain what is the stellar evolution. In that video, I will discuss different types of stelae found in the plants, in various plants. So stay tuned and enjoy my videos like this. Thank you.